you want to go?
Well, when I was about to interrupt, but I held back when Olivier was explaining what we really are is a gathering of, well, in the introduction comes that we're kind of a gathering place of curious interdisciplinary scholars from across the university. So along with Olivier, I'm one of the co-directors of the space. My personal background is in theoretical physics, but it feels like I'm getting farther and farther away from it into anything that's impossible, that there's impossible to conceive of how one might even construct a methodology to get there. Those are the questions that fascinate us endlessly. And so most of the community is comprised of students, but some recent graduates, but really from representation from all the faculties of the university and undergrad to postdoc, which is also quite nice because it gives a diversity. So the students or the scholars that are within the space, they kind of come with their own thing that they want to understand. And that's their project that they work towards. And whether it yields a concrete, tangible result or if it just yields greater understanding, all of it's okay.
Or more questions.
Or more questions, usually that definitely comes. And then, but then kind of interspersed with the programming are lots of discussions. It just places to think together. And that's when we find fascinating thinkers or people that we would like to talk to, we bring them in. And so for those 15 minutes, genuinely, you can speak about what's on your mind these days. You can speak about questions that you have right now. You can give the beginnings or the origins of your research journey. It's very much up to you, and we'll definitely come back to you with questions after.
So just to let you know, the projects at Building 21, what we tell students is the projects you do here, they must be project that you can do in the regular, more traditional programs. Not because the programs are bad or anything, but we want to provide students with the ability to do something that's overlaps many discipline or is ultra multidisciplinary or is very creative, was very embryonic, or maybe too large as an idea. Like I want to, we have a student this year who wanted to do something about Mars landing, right? Putting robots on Mars. We have another one here, Murad, who wanted to do created
Not wanted to do, he did.
He did large language models where they could actually, from your input, tell you a story about cities in the future. The problems. Emma here is wondering about randomness, so it's all across the board.
We have Mohammed online.
Yeah.
Fantastic. Yeah, that's amazing. When I became aware of your efforts, I thought, wait, this is fabulous. Just fabulous.
So I think what time is it in here?
It's 12:34.
Yeah, I think we can probably start.
Okay.
We have Lisa, Murad, Viola, Anita, myself, and Emma. And online, Darius is here, doing a PhD in neuroscience. Mohammed, I can see, is here also. And
PhD
Sean,
in bioresource engineering.
yeah, and Sean Manuel, I don't know who that is. This might be one of the students. So.
Right. Yeah, amazing. Well, thanks so much for having me here. I really look forward to discussing these issues. I'm happy to answer any questions afterwards or discuss whatever you want. I thought I would show you some unusual biology that people don't often think about. And when I talk to my own students, one of the first things I point out is that we have this beautiful textbook, let's say a developmental biology textbook. And what's at least as interesting as what's in the textbook is all the stuff that's not in the textbook. And why is it not in the textbook? And what can we make of it? So I'm going to, I'm just going to take 15 minutes. Usually I give a pretty long talk. So in 15 minutes, I'm just going to fly over a few interesting examples, and then you can all direct the discussion wherever you want. We can talk about any of these things. If anybody wants to follow this afterwards, this is where all the stuff goes that I have a pretty strong evidence for. So this is all the peer-reviewed papers, the software, the data sets, all that. This is the much more speculative stuff where I kind of talk about what I think it means. Okay, so the first thing that's important to understand is that a lot of philosophy, a lot of social science talks about the human, the various capacities, the rights, the responsibilities, and so on of a human. But one of the key things that biology is teaching us is that we sit at the center of several very dense continua, such that both in evolutionary time and developmental time, you can sort of very slowly and gradually run back the clock until you get to single cell microbes and even before that. And so, whatever we think of as true of this modern standard human, you have to ask yourself, well, how does that scale up from things that existed here? And in particular, if you think that there are any kind of sharp transitions or phase transitions or anything like that, you really have to have a story about it. And I'm not aware of any good stories like that that actually have a sharp phase transition. What I think of is a very smooth and gradual continuum. There's also now with technology, we know that we can modify these forms both in biological ways and in technological ways. A lot of this is already being done. And that further causes us to ask, what is actually essential about this? You know, what is really critical? Is it the genome? Is it the specific anatomy? Because all of those things are going to float. Well, they always did float, but they're going to float more strongly. And that's because at every level of organization, we can make chimeras. We can introduce engineered components into the relevant level of organization of life and ask what happens then. So really these continuous metrics are very, very important. And what I'm interested in my group is to develop a framework for understanding diverse intelligence. So we do all kinds of things, everything from fairly philosophical things to computer science to work that's preclinical in regenerative medicine, birth defects, cancer. But what ties it all together is my desire to understand embodied mind. So I really want to understand how mind exists in the physical world and the various manifestations of the intelligence of these kinds of minds that play out in very diverse spaces, which I'll talk more about that momentarily. So the idea is that when you are confronted with various continuum of systems that are quite different. They're not necessarily brains. They're not necessarily even alive. I'm much more interested in the category of cognitive than I am about the category of living versus non-living. And one way to start thinking about all of these kinds of categories and to try to be able to reason about and communicate with and discover and perhaps even create different kinds of minds and unconventional embodiments, we really need to think about what do they all have in common, because it isn't going to be what they're made of, it isn't going to be how they got here, meaning designed versus evolved. It's going to be something else. And so what I've been working on is this thing called an axis of persuadability, which basically holds that when you make cognitive claims about something, what you're really saying is, this is the protocol by which I plan to interact with that system. And that's a very testable thing, because you can go ahead and do that, and then we all get to find out how well that worked out. So the idea then is that when you look at these diverse kinds of systems, you can't have philosophical feelings about whether you think it is or isn't cognitive or intelligent or whatever, whichever of those terms you want to use. You have to do experiments. You have to have a principled science of being able to say, this is why I think this system is only amenable to certain kinds of relationships. So here, what you're talking about is you can only use physics and the traditional engineering, and then here these kinds of homeostatic systems you might use control theory or cybernetics or something like that. By the time you get up here, you've got some behaviorist theory, and in general, the tools of behavioral science, so training and rewards and punishments and things like that. And then by the time you get here, you really are talking about some very kind of advanced bi-directional relationships, psychoanalysis, love, friendship, whatever it's going to be. So in my group, we take very seriously the idea that where something goes on this continuum is an empirical question. You can't assume, you have to try. And in particular, we ask, where do groups of cells fit? So we treat development, regeneration, this kind of morphogenesis, when cells create complex anatomical structures, we treat that as a collective intelligence navigating the space of anatomical possibilities. And the reason that's important is because all of us were, at one point, what quote unquote just physics. And I hate that term, but people use it all the time. They'll say, I'm a real being with goals and hopes and dreams. That's just a mechanical system following the laws of physics. Well, guess what? We all, if you drill down far enough, we all follow the laws of physics. But something very interesting happens from here to here. This is a little blob of chemicals, but slowly and gradually through embryonic development, it gives rise to one of these remarkable forms, or even something like this, that's going to then go on and try to define what a machine is and say that we in fact are not machines, or maybe a very different kind of machine. But the fact is that developmental biology offers no bright line where you can hear this magical snap where you go from physics to mind. So what we really owe as scientists and philosophers is a story of scaling. We need to understand what are these things capable of, and how does it scale up to give rise to what this is. So we all make this journey, and we are made of an amazing material. We are made of an agential material. This is a single cell. This is a lacrimaria. You can see how much competency it has in its local environment and its tiny little unicellular goals. It has no brain, there's no nervous system, but it's really good at managing its metabolics, its physiology, its shape, and its behavior. And being made of this agential material means several things. I mean, agential material is engineered in a very different way than passive matter or even active matter. And both evolution and current bioengineers have to work very differently with things like this. The benefit is that when you do create large-scale and complex systems out of this kind of agential material, you get some amazing properties. I'm going to show you just a few examples. The first example is that I really like is this. So this caterpillar has to become a butterfly. Caterpillars are a soft-bodied creature. They crawl around in two-dimensional space. They have a little brain that's suitable for that. They have to turn into this. This creature is a hard-bodied kind of thing that flies and lives in the 3D space. So during this process, the brain is basically mostly dissolved. All the connections are broken. The whole thing is refactored and rebuilt into a new brain. Now, one basic fundamental thing is that if you train the caterpillar to find leaves on an appropriate colored disc, you will find that after all of this, the butterfly or moth still remembers the information. Okay, let that sink in. So the first thing we know is that kind of information survives this reassembly of the brain. But it's actually much more interesting than that. It's not just how do you keep information during that process. It's actually more than that, because the specific details of what the butterfly learned are completely, the caterpillar learned rather, are completely useless to the butterfly. In other words, butterflies don't eat leaves. The kind of motions that caterpillars do are irrelevant to the butterfly. What has to happen here is that information has to be generalized, not leaves, but food, and it has to be remapped onto the new body and the new lifestyle of the butterfly. That information, what it's preserving here is meaning. It's not preserving the fidelity of actual specific linkages between certain kinds of emotions and certain kinds of rewards. It's preserving the meaning of the deep lessons that it learned into its new higher dimensional life. So that's one example. This ability of information to be remapped in biology. It gets even more drastic here. These are planaria. These are flatworms. These flatworms regenerate their heads. Well, in fact, they regenerate any part of their body. Here you can cut them into lots of pieces. But what is found is that if you train these planaria to find food on a particular bumpy substrate, and then you cut off their heads, including their centralized brain up here, the tail will sit there doing nothing. Eventually, it will regrow a new head. And at that point, you find that these animals still remember the original information. So now the information is somewhere, presumably and throughout the rest of the body, but it's imprinted onto the new brain as a brand new brain develops. So you can see this. There's two kinds of information here: morphological information to how do I regrow my brain, but also behavioral information. What did I know? Movement of information from the body to the brain. So movement of information across tissue, and all kinds of interesting philosophical questions about what's it like to be a creature that can be multiplexed this way, where everybody remembers the original information. Okay, and the last example I want to show you is this incredible plasticity of these systems. So here's a tadpole. Here's the mouth. Here's the nostrils. Here's the brain, the gut, and the tail. So what you'll notice is something very, very weird about this tadpole. It has no primary eyes up here, so we've prevented those from forming. But we did put an eye on his tail. And what you will find is that eye will make an optic nerve. The optic nerve comes out here. Sometimes it lands in the gut. Sometimes it lands on the spinal cord and stops. It never goes directly to the brain, but sort of fishes around here for something to connect to. And these animals can see perfectly well. We built this machine that tests and trains them and then tests them on visual cues, so they can do visual learning perfectly well. So just imagine what this means. With no additional rounds of evolutionary adaptation, you scramble the sensory motor system. You provide this in a completely different configuration. and no problem, it can make it work. The brain that expected input from these two eyes for millions of years has no trouble somehow figuring out that this weird itchy patch of tissue on its tail is somehow producing visual data. So that that doesn't need novel evolutionary cycles to get to that. So what we have here is the body is, and in fact, it goes beyond individual bodies into groups and swarms. It's a multi-scale competency architecture at every level of organization. It's not just more complexity, but it's actually problem-solving competencies. All of these different layers are solving problems in different ways. If I had more time, I would give you all kinds of examples about how these different layers solve different kinds of problems. And the reason that's important for us is that we humans are really bad at detecting intelligence more broadly. We're quite used to recognizing intelligent behavior of medium-sized objects moving at medium speeds in three-dimensional space. So, you know, crows and parrots and dogs and maybe an octopus and maybe a whale, something like that. But it's really hard for us to recognize intelligence more broadly. All of our sense organs face outward. Just imagine if we had like this internal sensor of blood chemistry where you could directly feel, I don't know, 20 different parameters of your blood physiology. I think we'd have no problem then recognizing that we live in a high dimensional space where your liver and your kidneys are functional, clever agents that solve problems in physiological space. Though they take these different journeys in this physiological space every day to keep you healthy. So there are many kinds of spaces that we are now just beginning to understand. There's the space of all possible gene expressions. There's the space of possible physiological states. And then there's the thing that we study in most of all in our lab, which is anatomical morphospace. It's the multidimensional space of possible anatomical configurations. And I'll just show you a couple of examples of why this is interesting. So this is a human torso cross section. Look at this incredible order. Okay, everything is where it's supposed to be, the right size, the right shape relative to the right thing. And you might ask, where does this information come from? And of course, people are tempted to say it's in the DNA, but we can read genomes now, and really long before we could read genomes, it was already very clear that the genetics does not specify any of this. What the genetics specifies is the tiny molecular hardware that every cell gets to have. This is the result of the behavior of groups of cells playing out in various spaces. So the software, this is not in the genome any more than the exact shape of a termite mound or the shape of a spider web are in the genomes of these other creatures. So what we're trying to do is to understand that software of life. How do cells know what to do? How do they know when to stop? How do we communicate to them to induce them to repair something that's missing or damaged? And as engineers, we're also interested in how far can we push shape change? Could we get these cells to build something completely different? And how do we convince it to build something to get the hardware to build something very different from the default? And one way that we've begun to study this is to try to read the mind of the collective intelligence of these cells, literally to try to understand what are the biophysical mechanisms that merge multiple individual cells into a larger scale whole with a bigger cognitive light cone that can work on these grandiose construction projects instead of the tiny little projects that you saw that Lacrimaria doing. And it turns out that not perhaps at this point too surprisingly, although originally it was considered a pretty radical hypothesis, that it's actually bioelectrical communication. There are electrical networks all through the body, not just in your brain, that serve as the cognitive glue that binds individual cells into an emergent, coherent, larger scale whole. And we've learned to read and write some of these patterns. So I'll just show you what they look like. This is a time lapse of an early frog embryo putting its face together. And all of these bright spots that you're seeing, this is a voltage sensitive fluorescent dye that lets us see what it's basically like a brain scan. It lets you, except it's not in the brain, it lets you see all the electrical conversations that cells are having with each other. And this is one frame out of that movie. We call it the electric face, because this, in fact, is what determines the location of the different organs in the face. And if you change these patterns, you will change gene expression and you change anatomy. And this is how we can put eyes in weird places in the embryo and change the shape of the face and so on. So this is the normal this is the normal pattern memory that tells these cells what kind of a face they need to build and how to build it. This is a pathological memory induced by a human oncogene injected into some cells here, which will eventually form a tumor. But even before that, these cells disconnect electrically from their neighbors. So you can see they have a different electrical profile than all the normal cells. And once you do that, as far as you're concerned, you're just an amoeba and the rest of the body is just outside world. So what's happened is it's not that you've become more selfish, it's that your self has become smaller. So now you're no longer part of this collective network. You're just an amoeba and you go where you want and you know, do what you want. And that's metastasis. So we can now read these patterns, and you can imagine all sorts of therapeutics there. So just a couple of final thoughts. One is I wanted to show you these planaria. So these are these flatworms. They have some amazing properties. They're highly regenerative. They're very cancer resistant. They are immortal. So the asexual forms have no obvious aging to speak of. They just go on forever. They're resistant to foreign genes. So there's no transgenesis in the system, despite decades of us trying. Well, the whole community has been trying. And all of that, you might say despite, but I actually think because of, and it's a story we can talk about, an extremely noisy genome. Their genome it's very chaotic. You know, the cells have different numbers of chromosomes, they're mixoploid and so on. And despite all of that, they have this amazing property. And when you cut it into pieces, each piece remembers exactly what the shape is supposed to be, how many heads, tails, and where they should go. So we started to study this question: how does the body remember what it's supposed to look like? And we found this bioelectrical pattern here that indicates how many heads and how many tails. And we've learned to rewrite it. So here we've taken this animal, looks perfectly normal, one head, one tail. The molecular markers tell you this is the head, that's the tail. But what we've done is put in a false memory. So this is incepting a false pattern memory into the body of these animals that tell it to build something quite different if it gets injured. So this is not Photoshop or AI. These are real two-headed worms. And so when you amputate this animal, it consults the pattern. It sees that there should be two heads, and that in fact is what it builds. This is literally reading the memory of the collective intelligence of cells. It's also a counterfactual memory, because this two-headed form is not a map of this animal. It's a map of this perfectly anatomically normal one-headed animal. This is the representation of what you think a correct planarian should look like is not the same as what you look like now. That's what makes it a counterfactual memory. It's a representation of something that isn't true right now, but it will be true if you get injured and have the chance to regenerate yourself. I also call it a memory because if you cut these two-headed worms in perpetuity in plain water, they will continue to form two-headed animals. It's permanent. It's a permanent change. There's nothing wrong with their genome. Nobody has touched their genome. There's been no genetic editing, no transgenes. If you were to sequence it, you would have no idea that these things have two heads. This is not information you can derive from the genome, because that is not where the information sits. The information is kept in a bioelectrical circuit, which I showed you a moment ago, and it's rewritable. It's long-term stable, but it is also rewritable, like every good memory. So once we've turned it two-headed, it keeps that pattern as two-headed unless we change it back, and we do know now how to change it back. And so this is here. You can see what these two-headed worms are doing. And the final thing I want to show you is this. So here's a little creature running around, and I might ask what you think this is, and you could guess that this is something I got out of the bottom of a pond somewhere, right? So it's some sort of primitive organism running around. And I could tell you that if you were to sequence the genome on this little creature, what you find is 100% Homo sapiens. These are human cells. This is made of adult human tracheal cells taken from adult, often elderly patients. These cells, when given a chance at a new life, will self-assemble into a motile little proto-organism like this. We call them Anth Robots, and they have all kinds of interesting behaviors. We think this is the beginning of some personalized agential interventions, meaning that you could, from your own tracheal cells, make one of these guys and inject it into your own body for medical purposes. You wouldn't need immune suppression because they would be made of your own cells. There's no genetic modification here. There are no weird nanomaterials or any synthetic circuits or anything like that. These are your own cells. They share all the priors with your body about what health is, what disease is, what inflammation is, what cancer is, and so on. Now, why would you want something like this running around in your body? Well, because one of the things we found is that if you make a lawn of human neurons like this, and you put a big scratch through it like that, so a wound, this is how people study neuronal wound healing and so on. These Anth Robots can come down here. This is, we call this a super bot. So there's like, I don't know, 10 or 12 of these things. So they'll sit there in this cluster. And when you lift it up 4 days later, what you find is that what they've done is they've healed the scratch. They've taken the two sides of the neural wound, and they've healed them together. We have no idea why they do that. This is the first thing we tried. So my guess is there's probably hundreds of other useful things they do. This is just the kind of the first thing we found. But who would have thought that your tracheal cells, which sit there quietly in your airway for decades, if given the opportunity, could self-assemble into a little creature that does something completely different, has a different behavior, different morphology, doesn't look anything like a human embryo, and yet is able to do some interesting things like this. And so the final thing I'll say is that, when Darwin said endless forms most beautiful, he was really inspired by the richness of the variety of the standard life forms here on Earth. And I would like to point out that that is probably a tiny little dot. All of it, everything, all the amazing creatures that we see here on Earth are a tiny little dot in the enormous space of possible bodies and minds, because biology is incredibly interoperable with other things. We can talk about why that is. And therefore, almost every combination of living material, engineered material, and software is some kind of new agent. So hybrids, cyborgs, various chimeras, all of these things are possible. They will have some kind of body, they will have some kind of mind, and they will, all of our old heuristics about how to relate to other beings based on where on the tree of life with us they are, all of those things will crumble. And for you young folks, over the next few decades, this is the world that we will be living in. We have to update our ethical frameworks to be able to much more deeply understand how to relate to beings that are completely different from us. So a kind of ethical synthbiosis, I like to call it, with other creatures, where it's going to go far beyond the old kind of ways, which simply ask, what do you look like? Meaning, what are you made of? And how did you get here? Were you engineered or naturally evolved? Those questions are going to be useless very, very soon. So that's it. I would just like to thank here some students and postdocs who did the work that I showed you today. Lots of cool collaborators, and of course our funders. And I have to do a disclosure. There are 3 companies that have spun out of our lab that support some of our research. And always thank the animal model systems, because they do all the hard work teaching us about biology. So yeah, thanks. I'll take questions.
Okay, I think we're absolutely stunned.
Yeah.
We have tons of questions. Okay, so I'm going to ask stupid questions because I'm not a scientist. I'm a literary person. When you mentioned the cognitive glue, right, the electricity, how is that? Is that cognition? Is that a vector to cognition? What's the relationship between that and the actual working of the neurons?
Yeah, so a couple of things. I don't think you need neurons for cognition. I think neurons are a good way to do one kind of cognition. Neurons are great if you're going to solve problems at a very fast rate of speed by running around in 3-dimensional space, which obviously once muscles and brains came along, that was something that animals like to do. But there are lots of other problem spaces in which very unconventional beings live and work and suffer and so on. And there are many kinds of cognitive glue. Bioelectricity, whether among neurons or among the rest of your body cells, is just one kind of mechanism. And what I mean by cognitive glue is it's just a set of policies that allow a collective emergence of a larger cognitive light cone, where the system becomes more than the sum of its parts, and it's able to pursue goals that are larger and larger. So I'll just give you a very simple example. If you have a rat and you've trained the rat to press a lever and get the reward, well, the rat knows that the lever is associated with the reward, but there's no single cell in that animal that had both experiences. So the cells at the bottom of the foot touched the lever, the cells in the gut got a delicious reward. No individual cell could possibly own that associative memory. And so you need something like a cognitive glue to bind those cells together into an emergent, coherent self, which is the owner of this kind of associative memory that no individual cell can have. And so that's the standard story of neuroscience, is that electrical networks within the body are the kind of thing that allows that. More broadly, there are other principles that we can talk about if you want to dive into it, about what it is that allows individual subunits to scale up to have goals that are bigger than any of the systems themselves can have, right? So that scaling of the cognitive light cone is what you're looking for. This is what we call life, or things that are good at scaling their cognitive light cone beyond that of their parts. And electricity is just one of those things, but it's a really powerful one.
Mohammed, do you have a question?
Hello. Thank you very much for the presentation. It's very interesting.
Thanks.
I have two questions. I guess, have you done any research on the implications on like evolution? You're saying there's a lot less influence from genetic information and it has more to do with the bioelectrical signals. So I guess, have you done any I guess research into what that means towards evolution? I guess because the Planarian worms, you said the changing of the electrical signals was permanent. So every time you split them, each new worm had two heads, right? So I guess, could you speak a little bit on that?
Sure. Yeah. There's something very interesting that goes on when you ask about how evolution would work on a gential substrate. So typical standard stories of evolution are really told from the perspective of searching for configurations of a passive material, but that's not what biology is. Real evolution searches through what it's really searching through is the space of behavior shaping signals by which cells can hack each other. And when you incorporate that layer of competency, of that developmental that morphogenetic layer between the genome, which is what gets mutated, and the function, which is what gets selected on, that layer it changes the whole dynamic. And I'm going to tell you a quick story about how that works. Imagine that you're a tadpole, you're a frog, and imagine what would happen if the frog was a very a kind of hardwired kind of system where the genetics determine exactly what the shape is, and that's it. If that were the case, imagine you have a mutation, and that mutation moves the mouth off kilter a little bit, but it also has some really useful implications somewhere else in the body. Well, once your mouth is off to the side, you're not going to be able to eat, and you're dead. And evolution cannot examine the possible beneficial qualities of this other mutation. So that's, when as an engineer, when I first, I was very young, and I first heard of evolution and this idea that, well, what we're going to do is we're going to take these complex systems and make random changes. I was like, what, that's never going to work because for obvious reasons, right? If you ever tried to change a complex system, of course, most of the time you make it worse, not better. But the real situation is quite different. Actually, in the tadpole, what we discovered a few years ago is that if you actually scramble the face of a tadpole, we call them Picasso tadpoles because everything's in the wrong place. They actually will rearrange all the organs and get back to a normal frog face. So that mouth that's off kilter will actually get back to where it needs to be. And this is it's not just about the frog face. This is all over the place. Early human embryos, you can cut them into pieces and you don't get half bodies. You get monozygotic twins and triplets and so on. So that repairability, that competency to get your job done despite all the weird things that can happen to you, which by the way is a definition of intelligence, that ability does something very, very crucial to evolution. So imagine, you've got this tadpole and everything's working fine. And so selection looks at that tadpole and says, okay, this is a great tadpole, but here's the problem. You don't know if you're a great tadpole because your genetics was fabulous, or you're a great tadpole because your genetics was actually not very good. Your mouth was, somewhere else, but you fixed it. So your competency is good. So the problem that happens when selection can't see, can't distinguish those two cases is that it has a very hard time improving the genome. Right? Because you can't see the genome. You don't know which ones had the best genome because all of them look pretty good. So what we found, we did these simulations, and so we did a very in-depth computational study of this. And what we found is that when that happens, evolution spends all of its time improving the competency. And so now you've got this positive feedback loop, because as soon as you improve the competency, that makes it even harder for selection to see the genome. And so the competency goes up more, but then that makes it even harder. And so you've got this positive feedback loop that if you let it run all the way, you end up with Planaria, which Planaria, basically all the effort went into the algorithm of creating a perfect worm, no matter what the hardware looks like. The Planarian genome is actually pretty junky. And that's because, unlike the rest of us, when we reproduce, our children don't inherit the mutations that occur in our bodies, right? That you have a baby from an egg and a sperm, and whatever happened to your body is generally not passed on. The mutations are not passed on. Planaria don't do that. When they reproduce, they cut themselves in half, they split themselves in half, and then they regenerate. That's how they reproduce. So that means they keep all the mutations that don't kill a particular cell. So they've been accumulating for 400 million years. They've been accumulating these mutations. That's why their cells have different numbers of chromosomes and all that. And yet, this used to, we only figured this out like a year ago. Before that, for the last 26 years, it drove me insane that why is it that the animal with the worst genome is the one that's highly regenerative, cancer resistant, and immortal, right? That is not the story you're told in your biology classes. What you're told is that your genome is really important, and that the genome determines everything, and therefore the animal with all those properties should have a rock solid genome. It should have like the most pristine genome ever. And that's exactly backwards. And it's backwards because what evolution did was to create a system that assumes from the beginning that the hardware is going to be junky. You know from the beginning that you cannot rely on your hardware. And there are many, many examples of that. There's a whole hour talk that I could give on that, just to talk about all of the things that biology does not assume are going to go right. Because unlike our computer technology, which is meant to be very reliable, data is meant to stay where it is, the things are not supposed to break. Biology is completely unreliable at the lower levels. Everything is going to break. Everything, there's error, there's noise, everything is sloppy. But when you assume that that's the case, you build algorithms that don't require these micro level details to be right. And that's what happens in Planaria. In Planaria, that ratchet went all the way to the right. Like we're just going to assume the hardware is junk, and we're going to focus all our efforts on an algorithm that creates the right thing, despite our parts. And then there's sort of in between cases. So salamanders are pretty good at that, but not as good as Planaria. So salamanders do age, and they don't regenerate everything, but they regenerate many organs. And below that, you get mammals where the embryos are pretty regenerative, but after that, no. And below that, you get nematodes, for example, which they don't, they're like cookie cutter organisms. You like all the cells are numbered, like exactly which cell is where, and that's it. That's all you're ever going to get. So that ratchet. So that's what happens during evolution, is this intelligence ratchet that gets cranked up by having an unreliable substrate. The unreliability of the substrate, the fact that evolution knows that everything is going to be different. You're going to be mutated. The environment's going to be different. Your own parts are going to change. Everything's going to change. That's why I think what we have here is a system that doesn't make solutions to specific environments, right? That's what people are taught a lot, is that evolution will select a good solution that's a fit to a specific environment. I don't think that's what's happening at all. I think that what evolution is doing is creating problem-solving agents that are good at solving problems despite novel environments. And that's basically intelligence. And the intelligence required to deal with an unreliable material is what gets then spun up into this amazing morphogenetic robustness, like in Planaria, and eventually into behavioral intelligence that we all see every day.
Thank you very much.
Okay, so again, fascinating. I absolutely am fascinated. I was about to say I'm without words, but obviously I'm with words. Where does that model of that perfect model is coming from? Like, so you say you scramble these animals, they have this Picasso face, but then sort of the intelligence of the model, the competency brings it back to its rightful place. Where does that model comes from? Is that, and how does evolution decides that mouth should be here instead on the side? And because it seems as if this like almost this Platonic ideal of how what an organism should look like. Where does that come from?
Yeah, I mean, so this is a very strange thing for a biologist who does biophysics at the lab to say, but I think you put your finger exactly on it. And I'm now starting in the last about a year or so, I've been actually starting to talk about Platonic space and the notion of these xenobots, Anth Robobots, all these weird synthetic things we're making as tools, exploration tools to explore Platonic space. I really think that's what this is. And obviously this is, you know, just so everybody knows, this is not the standard story that you'll get from you know, my molecular biology colleagues. You know, this is considered really crazy kind of stuff. But I actually think in the end, the answer to that is the same answer, which is what Plato and Pythagoras were going for when asking where the truths of mathematics come from. And the way that I usually illustrate this to students is that you've ever seen a Galton board? You guys know what that is? It's like a just imagine a vertical piece of wood, and what you do is you bang a bunch of nails into it like this. You could buy this toy on Amazon, so like 20 bucks. You bang a bunch of nails into it, and you take a box of marbles and you dump it in, and the marbles go boom, 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 and they land. What's the shape of the marbles, right? They make this beautiful Gaussian distribution. So you could ask the question, wow, where is that shape encoded? So I'm going to study the marbles. I'm going to study the wood. I'm going to study the nails. I'm going to measure the, you know, the distribution of the nails in this thing. And after you do all of that, you are no closer to the question of where is where does that shape come from? So what the machine is doing, what the physical body of that machine is doing, is it's pulling down some laws from, you know, there are laws of computation, laws of mathematics, there's a million different kinds of things like that. And I think what biology does is create bodies that are pointers into that space. And so what I want, one research agenda that you could have, and that I think this is what we're doing, is to create different types of bodies that pull down different types of affordances from that space. And thus you can start to map out what's going on there. So the standard embryo or the standard shape is like this one pinpoint that you normally see, and it's quite reliable. But if you've got the right set of tools and the right research agenda, you can start widening that hole, and you can start looking at the space of the latent space around that. And I think that's exactly what evolution does, is it makes creatures that navigate that space by pulling down specific forms. And I can't begin to tell you yet what else is up there, but the fact that, I mean, these I haven't even showed you the Xenobots, but Xenobots, Anthrobots, all those things, they've never existed before. They have shapes and behaviors that you cannot say they're due to eons of evolutionary selection. There's never been any Xenobots or Anthrobots. There's never been selection for those things. Where do those patterns come from, right? Because the standard answer to your question would be, well, evolution, meaning that for you know, you're a froggy shape because for years everything that wasn't quite the froggy shape died out. That's why. And that's great, but that's not a good explanation for any of these synthetic things that we make, because they've never existed before. So that's what I think evolution is actually doing. It's developing pointers into that space, and I think we need to understand the structure of that space.
Other questions? I've got tons of questions. Go ahead, Anita.
Well, so everything you say is fascinating. I have a question that's been playing from the get-go, but I sort of want to let the biologists go first because it's not my field. But now that I'm speaking, I'll go. In the beginning, it sounded like you're kind of alluding to, there's no evidence of like the emergence of a complex intelligence. It just sort of, it seems that we accept this smooth transition of, you know, a single cell has its own kind of intelligence, and then groupings of cells have a particular collective intelligence, and then it brings itself up, and suddenly you have the human. But I just wonder if you can talk more about, have you looked at these boundaries, and do you have any insight into, you haven't seen any sharp transitions, but do you have any insight at all into these boundaries?
Yeah, so what I think happens is that, yeah, this is our research agenda. So we have to understand how the scale up is happening. And so we've seen them. I don't think there are any boundaries per se. I think there is a gradual the thing that's very gradual is the enlargement of the cognitive light cone, meaning the scale of the goals that a system is able to pursue. All right. And then the thing that does maybe look like a phase transition, I'm not sure yet, is that cognitive light cone gets used in different problem spaces. So for example, I'll just give you a simple example. Well, first let's talk about the cognitive light cone. So if I ask you, what are the things you practically care about? What are the things that you actively work to maintain? And you tell me, well, I am only interested in the sugar concentration within a 20 micron radius, and I can remember the sugar concentration back 5 minutes ago, and I can predict the sugar concentration forward 5 minutes. I'm going to say you're probably a bacterium. That's a very small scale set of goals. If you tell me that, well, I have a couple of hundred yards that I'm in charge of, and there's no way I care about anything that's going to happen 3 weeks from now, a couple of miles over, you might be a dog. That's the level of cognitive light cone that you can handle. And if you tell me that, well, I'm working on the state of the financial markets 100 years from now globally, I'm going to say you're probably a human because you've managed a cognitive light cone that's bigger than your own lifespan. And if you tell me that you care about, in the linear range, you actually care about every sentient being on Earth, or even more than a couple of hundred beings actually, then I'm going to say you have to be beyond a human. You are some sort of bodhisattva-like creature that can actually care about that many beings, because humans can't really manage it. So that's the first thing. So the cognitive light cone is just the scaling of goals. Now this we can see, because we can see how this works, because if you have individual cells are homeostatic with respect to physiological goals. So they're trying to maintain pH, hunger level, all kinds of things. And you can see there are these little loops that take measurements and make adjustments. When you take a bunch of these cells and you join them into an electrical network, a couple of interesting things happen. One interesting thing that happens is that the cells have a little bit of a loss of personal identity. And what I mean by that is, imagine that you have two cells and you're connected by this electrical synapse known as a gap junction. That means that when something happens to cell A and there's an event, let's say a calcium flux, that it serves as a memory of that event, that propagates through the gap junction to cell B. Cell B has no way of knowing, wait, that wasn't my memory. That was somebody else's memory. I mean, a calcium flux is calcium flux. You don't know who it belongs to. So instead of saying, I didn't get injured and or maybe he got injured, you're saying, well, we got poked and it was in that direction. So what it does is, by wiping the memory metadata on experiences, it allows for a shared history among a collective of these cells. They have now a much bigger spatial area that they're working on, but they also have a much bigger temporal area and computational capacity. And the individual cells can't do the calculus that amoebas normally do, which is every man for himself. They can't do that anymore because they're sharing the. And we've actually done experiments on that too. So what you have is you have the scaling, the spatio-temporal scaling, because the whole network now can remember much larger things. And one of the things it can work on is morphogenetic goals. It might say, what I'd like to do is to minimize the curvature along the cell sheet. Individual cells won't do that, but the group will say, well, you know what? Yes, we need to minimize the tension or something like that, and you start to get morphogenesis. So they have much larger goals. So what you can see here is the scaling. That's why I like this bioelectrics, just because it's easier to work with. I'm sure there's other mechanisms that we don't know about, but they allow the kind of mind meld among the cells that enables cooperation. It enables larger memories, larger goals, larger computational capacity, and then it enables some other things. One of them is stress propagation. So just imagine if I'm a cell at the bottom of this thing and I need to be up here, I want to go up here, but there's all these cells in my way and I can't get up there, and they're all happy. They're not going to let me through. One of the things I could do is and I'm stressed out because the stress is it's a generic measure of delta of error, or how far away are you from your goal, right? So I'm stressed out. I start to imagine, if I start to secrete a stress molecule, I'm going to stress out everybody around me. They don't know that it's my problem, not their problem. It's the same stress molecule. So now the temperature in the sense, not physical temperature, but like simulated annealing kind of thing. The temperature goes up. Everybody gets a little plastic. They're all willing to do some things different. Now I can get up where I need to go. Then everybody's stress goes down. So that stress sharing basically makes my problem becomes your problem, not because you're altruistic. It's because the mechanism facilitates cooperation towards solving these kinds of problems in a collective way. So all these kind of dynamics are what, and there's a few others, but these kind of dynamics, that's the cognitive glue. That's what allows little tiny intelligences to become a bigger intelligence that cares about things that the individual cells have no ability to recognize.
Thank you. Fascinating.
Emma?
I have maybe a more simple question, but I'm very curious about the experiments you ran where you created one with two heads, and I'm curious as to how you induced the electrical current to that matches the head, because I don't really understand how it works. I'm not sure if there's an actual pattern of the electrical current, or if it's just about the place. But if it's about the place, then I don't see how putting activity at the tail would induce a head. So yeah.
Yeah, okay. I'm gonna try to describe this verbally, because the real way we explain all this is with a mathematical model of a bunch of coupled cells. All the cells have ion channels and gap junctions, and you can actually see how this circuit works. I'm gonna try to do it in words. So there are no electrodes. We don't apply electric fields. We don't do anything like that. What we're doing is hacking the interface that these cells are normally using to control each other, which is we control the ion channels. So on the cell surface of these little proteins, they pass ions in and out, and that results in an electrical pattern. What we have to do is we have to figure out which ion channels would we open and close, and there's many different ones, potassium, sodium, chloride, there's hundreds of different ones. Which ion channels would we open or close to induce the whole system to settle in a particular pattern that we want? This, by the way, is the holy grail of bioelectric medicine, because again, whether it's a tumor or a birth defect or whatever, the whole goal is to say, this is the correct pattern I want. What channels could I open and close so that the sheets, the collective of cells settles into that pattern? So it's a computational task. We have simulators that we use, and the reason it works, so there are two key things for the circuit. Remember that what you have is a bunch of coupled reservoirs. These little cells are coupled, and every cell has channels that are themselves transistors. They're voltage gated current conductances, right? They're voltage gated ion channels. So the capacity for all kinds of complex symmetry breaking and a spontaneous self-organization, right? It's an excitable medium. There's all kinds of stuff that can go on. So your job is to calculate what stimulus do I put where, and it's, by the way, very brief. It's only about 48 hours that you have to do this. After that, the system organizes and takes over and stores the pattern. And what I want, if I have the left end is, let's say the head end is depolarized and the tail end is not, what do I do so that this part becomes depolarized as well without messing up this part? So we found it. We found the way you can do it. There's a couple of ways you can do it. One way targets the gap junctions. Another way targets proton levels or potassium levels. There's a few different ways to do it. And what it ends up doing is it ends up creating this when everything settles out, it creates these two areas of depolarization. And that pattern is interpreted by the other cells as make two heads. That's the key, right? We're not micromanaging these cells. We're not telling each cell what type of cell to be, or that we're not talking to the stem cells or the genes or anything like that. We activate the very high level subroutine call, which basically just tells everybody two heads is the way to go. And that is the basic architecture of neuroscience as well, where when we try to do something, you don't think about which muscle fibers are going to go where. You have high level goals, and those goals get transduced into, talk about mind body interaction, right? You have these high level goals in the morning. You got to get up out of bed and go meet some social goals or whatever. That intent has to move potassium and calcium ions across cell membranes in your muscles. That's what your whole body does, is transduce from these incredibly abstract high-level goals in some crazy linguistic social space down into physically pushing ions around your membrane of your muscle cells so you can get up and walk and go do that. So that's what's happening here. We're providing a signal that basically is just a stimulus that causes the electrical pattern in the excitable medium to rearrange. And we found the one that rearranges in this way. There are other, we can do other things that way, but that's the one we found here.
Thank you.
I had a question kind of adding on to this. Once the worms have two heads, what's their behavior? Is there one head that ends up acting more or less as a tail, even though it has the shape of a head? Do both heads try to lead the organism? What does it do?
Yeah, that's a great question. And by the way, there are human cases of this as well, right? So there are some fascinating clinical examples where people have multiple heads with different opinions, and they have different personalities, and they can have you can have questions about sometimes they feel certain things together and sometimes they don't. there was a pair of conjoined twins like that recently, and one of them got married. You know, but they have different personalities. You know, the two women have different personalities and so on. So there are human cases like this, but in the Planarian case, what happens is at first the two heads kind of fight, and they don't really get it, you know, they don't have good control over the body because they're trying to do different things, and this makes them actually challenging to feed because they have a hard time feeding because they keep, you know, moving each other. But in some cases they figure it out, and in some cases they learn to cooperate, and then they sort of so you can imagine, right, if you've got two heads like this, you might be confused, but if this guy bends to his right and this guy bends to his left, you make a U and you sort of cruise around like that. So sometimes they can do that. It's pretty variable, and you know, it's hard to do those kinds of experiments in Planaria, so I don't have a good quantification of the behavior. It would be nice to do things like train one head and find out if the other head inherited the information, or whether they have higher IQ, right? Does having two brains actually give you a higher IQ? Lots of questions like that probably can be done, but it's really hard. We don't have good protocols for it yet.
Thank you.
So I think it's John, you have a question?
Yes, I did. Thank you very much. And just quick introduction, because I think Olivier, you did not know me. I'm a colleague of Darius's. That's why I'm here today. Thank you, Professor Levin, for this great talk. I'm a longtime fan of yours. I've been brushing up on your previous talks before this one, but just off of not this last person's question, the one before. I think you were pointing towards this giving a rather coarse signal to attain your specific goal, and then everything that happens between the signal and the goal is various levels of like interfacing and transducing, translating, something like that. And I think a implicit or maybe explicit point you're making is that we don't have to be worried about all of those specific steps. We can care about, ideally, you know, the minimal amount of those like translating steps so that we can attain the goal at the end, right? But then I think this is interesting from like an applied perspective, right? So for example, I think a lot of your main missions is regenerative medicine. It's very, you know, concrete, right? But then from a fundamental perspective, and then the reason why this is of interest to me is because I'm from neuroscience, and you spoke kind of to that, is from the fundamental thing. So like if I was able to give some kind of coarse signal to an individual or to a brain that attains some kind of goal, then obviously everybody would want to know, how did that work, right? They would want to be able to trace it, and they would not be satisfied by appealing to you know, into agential materials, transducing the signal in intelligent ways, you know, like they would want something else, right? And I wonder what you think of this when you know, you showed us the electric face, and the first like idea I guess that I had is, when I look at that electric face and when I imagine the types of interventions that you guys in the lab are doing on, I guess, an image, is that kind of like similar to like, if in neuroscience, if you're looking at like multivoxel pattern analysis of a brain or something, like, is it the same level of coarseness where you're like at a super like high level of what the substrate is doing, but you're satisfied with the result, and so that's it? Or do you think that there's like some kind of like fundamental difference between this like neural perspective and the bio one that your guys are doing, where you have increased confidence in like the actual like mechanistic explanation of what's going on? I don't know if that makes sense. Maybe you have thoughts on that.
It makes perfect sense. I mean, you're asking about the nature of causation and what a good explanation is, right? So I mean, really deeply, right? That's you know, when you say, will you be satisfied with that? That's the question. Is like, what's a good explanation? So it's a great question, and here's how I think about it. I mean, simply, yes, it plays out exactly as in neuroscience, and you can, and we have tracked down all the molecular steps. So we know when we make specific bioelectrical patterns, we know what the transduction mechanism is to change various gene expressions, what happens after that, what the second messengers are. We know all that, and so you have to track it down, you know, as you say, for other people to be happy about the state of things. But let's understand what that does and doesn't do for you. What we can, with the story that we can tell with a very high degree of molecular resolution, is how a single cell gets from having its voltage changed to the genes that will then turn on and off, and what happens to it after that. Okay, that story makes it very difficult to do regenerative medicine. It's okay if you want that story, we have it, But with that story, you're not going to make the kind of changes that you want, because large scale changes are not about tweaking individual cells. It'll be completely impractical. And it's a little bit like, if you imagine that you have this like amazing argument for somebody to I don't know, convince somebody to go to graduate school or something. Right? You talk to somebody and boom, and they're convinced. I'm going. And so somebody says to you, "How did you do it? What did you say?" And so what you give them is this incredible list of all of the velocities of all the air molecules that you pushed into their eardrum. That's the description of our conversation. Like, fantastic, and it's not wrong exactly from a physics perspective. I mean, yes, that is what happened. How useful is that to you the next time you need to convince somebody else to go to grad school? Right? That is not the core of what you're doing here. And so yes, we can tell stories like that, but what I'm interested in is the most functionally efficacious story. And sometimes it will be the molecular story. There will be single gene diseases and, certain stem cell kinds of applications where, yeah, it's just, you crank up, a Wnt11 and you're good to go. Sometimes that will be the answer, but a lot of times that will not be the answer.
Thank you. If I can quick follow up on that, is it possible?
Yeah, real quick, because it turns out it's already 1:30. I've got my grad student is texting me where I am.
Oh, well, apologies. I'll let someone ask a question if they want to go.
So
anybody else?
Any other last question, burning last question? Because I've got thousands, but Lisa?
Yes, but it's 1:30.
One last quick.
Okay, well, yes. What I was wondering is when you talked about the electrical patterns when you were talking about the frog face and how I guess those ion channels do their thing and determine the frog face, you said, to my understanding, it's not the genome that's, it's not the genetics, it's those electrical patterns. I'm just unsure where they're coming from. Is there any way you can talk on that, or maybe there's something I've missed that?
Yes, sure. No. That's a key question. It's a combination of several things. What the genetics does establish is the properties of the ion channels. So the genetics are telling you that you've got ion channels that turn on at a particular voltage, or they pass potassium versus chloride. The genetics sets the hardware. Once you've set the hardware, just imagine somebody gives you a description of how to build a couple of transistors, and with those transistors you make a logic gate, then you have this nice truth table that defines the AND function. And you say, okay, so where is that truth table encoded? And you're staring at your silicon and your germanium and the copper, and that's not where you're going to find your truth table. And so to be more specific, look up things like Turing patterns and just self-organized patterns in general. It's a combination of the genetics sending the boundary conditions, which is the hardware, and then that hardware exploiting the laws of physics and computation, which give you not just emergent complexity. Emergent complexity is easy. They actually give you emergent kinds of intelligence, emergent goal directedness, and that's the scaling that we're interested in.